Hi guys, welcome to lesson number 3 of week 1. In this lesson we're going to talk about this term's assessment task. As always, if you could do the do now questions, pause the video here, have an attempt. I'll put the answers on in a couple of seconds. Okay, here are the answers. Remember, we'll go over this together live via Zoom, but hopefully this makes sense. If not, maybe review the last video, your last notes, and we can talk about it at the Zoom. Okay, so I need to mention the assessment task. I know it's a necessary evil, something we just need to do, but don't worry about it. I'll explain exactly what I expect. This term is obviously a bit of a strange one, so the assessment task is going to reflect that. It's going to be slightly different from what I'd maybe usually ask for. So hopefully it might actually be a little bit easier for you to do, okay? So your assessment task for this term is to gather secondary source data to investigate and assess the evidence that supports a scientific law or a scientific theory, including but not limited to the theory of natural selection, the theory of plate tectonics, Big Bang theory, Ohm's law, or Newton's second law. So I'll explain this in more detail in this lesson, but what you need to do is essentially pick one of these and assess what evidence there is that supports that law or that theory. And I've put them there, including but not limited to. So these are the 5 that I would suggest you do. So you just need to pick one of these. You could pick natural selection, you could pick plate tectonics or the Big Bang theory. You pick one of those, and that is what your topic will be for the assessment task. Now, if there's something that you really want to look at that is not on that list, then get in contact with me and we'll discuss if it's feasible or not. So if there's something you really want to do that you maybe have an interest in or a lot of background information on already, then you can choose that topic if you discuss it with me first. And we'll say yes or no, okay? I don't want people saying, oh, I'm just going to go and do something different, and there's not that much information on it, or it's slightly off topic. So if you really have a passion for something that's not on that list, by all means get in touch with me at this lesson. So you need to be in the Zoom and let me know, and then I'll assess whether or not we can do that for your assessment task, okay? So your task is to gather secondary source data. I'll explain what secondary source data is in a little second. What you need to do, what I expect you to do for this assessment task is, number 1, be able to outline the scientific law or theory that you're studying. Number 2, describe the evidence that supports this law or theory, what experiments have been conducted, what results are there, what observations have been made. Number 3, assess the evidence. Based on your collected information, make a judgment on how well does the evidence or the observations that have been made support your theory or law. And then the fourth one, in your assessment, you must again explain why you have made this judgment. So why do you believe that the evidence that has been presented for this theory or law is acceptable? So first, I've said secondary source data quite a bit. What the hell does that mean? Well, primary data and secondary data are two different things. Primary data is information that you collect from the original or first-hand research. So, for example, surveys, focus groups, or experiments that you conduct yourself. So this image here in the top right with students doing some chemistry, they're collecting primary data. Okay, they're the ones collecting data. Therefore, secondary data is information that has been collected by somebody else, and you're using that information. So that's what you're going to be looking at for this assessment. So you're going to go online, find some books, any information that you can on your topic, and you're going to use the information that was collected by other people to present your case. So what do you need to submit to me? You must create at least a 5-page PowerPoint presentation that addresses each of the points, and I'll break that down again. And you must also provide Martin with a 1-page A4 fact sheet. So I'm going to give you an example of what it is I expect using germ theory later on. So I'll break down what it is at the very minimum I expect you guys to submit into me. So you're going to have until week 6 to do this, and you're going to have 5 lessons to complete this. So when you have that timetabled lesson, we will have an hour on Zoom. You guys can use that hour to do your research. That's more than enough time to do each part of the components. And if you have any questions or queries during that hour, I'll be available online to talk to you. So all of this can be completed during the allocated class time. If you want to spend additional time after class, that's fine, but you should be able to complete this within the allocated class time, okay? So you're going to get one lesson, an hour, to conduct background research and outline your theory that you've chosen. You're going to get another full lesson to conduct research on the evidence that supports your theory and law, and that will hopefully make up slide number 2 of your PowerPoint. You're going to get another full lesson to insert data or diagrams that support/explain your theory or law. That'll be your 3rd slide. You'll do another full lesson which will allow you to do research on the evidence. How well does that evidence back up your theory and law? And why is your theory or law important in the modern world? That'll hopefully be slide 4 and 5. And then finally, you're going to complete an A4 fact sheet that you'll submit to me with your PowerPoint presentation. Okay, so you're going to get 5 whole lessons to do this assessment task. And if you've got an idea about what it is you want to do already, then you could probably do this fairly quickly and do it well, and use that class time to really ask questions of me and make sure that your PowerPoint and your fact sheet is the best it can be. So here's an example. Okay, I'm going to use germ theory as an example. So germ theory, so say this is my assessment task. Germ theory is something that's really important to our understanding of how diseases work. And my first slide outlines what germ theory is. So germ theory, the definition, is the theory that certain diseases are caused by the invasion of the body by microorganisms, which are organisms are too small to be seen except through a microscope. So that's a nice little definition there. So I've explained to you what germ theory is. I think that's probably a really solid place to start with your own assessment task. What is it we're looking at in a nutshell? The French chemist and microbiologist Louis Pasteur, the English surgeon Joseph Lister, and the German physician Robert Koch are given much of the credit for the development and acceptance of the theory. So Louis Pasteur here, and your other 2 guys, Lister and Koch, who are really important. I put Pasteur at the top there because he's actually been really important for a bunch of other things. Have you heard of pasteurized milk? Pasteurized milk is named after Louis Pasteur, and pasteurized milk is called pasteurized milk because he came up with a method that would help sterilize milk and make it safer for people to drink. So Louis Pasteur is a really interesting guy, but he's also really important in the development of germ theory. What predated the theory before germ theory? So what did people believe caused disease and germs before? Well, it was called the miasma theory. The miasma theory was the predominant theory of disease transmission before germ theory took hold towards the end of the 19th century, and is no longer accepted as a scientific theory of disease. So remember, this whole term is talking about theories and laws that have been around and have maybe changed or been strengthened by evidence, or in the case of miasma, entirely dismissed based on the evidence that we now have. So miasma, this little image here, was basically the depiction of how people thought disease spread back before germ theory. And basically miasma theory was that dark air or night air, so air was carrying diseases, which, makes sense in a way. Air can transmit diseases, but it's not the only way. And it's not the air that is carrying the disease or causing the disease, it's the microorganisms within the air. So for example, infections that are airborne can carry through the air and cause disease. So if you didn't know that the microorganisms were causing that, it makes sense to think, okay, maybe the air is what's causing the disease. So the miasma theory was the predominant theory, but that changed as new evidence came to light. So what new evidence came to light? So this is my second slide. Evidence to support germ theory came through a lot of laboratory experiments. So laboratory research by Louis Pasteur in the 1860s and then Robert Koch in the following decades that provided the scientific proof for germ theory. Their work opened the door to research into the identification of disease-causing germs and potential life-saving treatments. So this experiment here has become quite a famous experiment in evidence for germ theory, and it's quite simple. It's quite nice to look at. This diagram explains it quite well. So number one, they have a solution that has microbial growth in it, and they boil the solution, and you see that they have this kind of fancy flute almost on the end. And what they would see, this experiment was, if they boiled the solution and waited, there was no growth. There was no regrowth of the germs or the microbial growth. If they did the second type of experiment where they boiled it again, but they broke off the neck of the bottle, air could get in and reinfect the liquid here, and there would be more microbial growth. So what they were showing here was when something is boiled and not re-exposed to the air, then it wouldn't be infected again. If something was re-exposed, it would be infected. And why? That might seem really simple and obvious to us now. One of the main theories of the day was that things could spontaneously grow, okay? But without the access to the air again, this experiment demonstrates quite clearly that things cannot grow unless there is further exposure. So that's a really simple experiment that makes sense to us right now, but it was a really, really important one to give us the first indication that regrowth of microbes could only happen after exposure to, say, the air. So Robert Koch came up with a bunch of postulates, and they are called Koch's postulates. He postulated, so a postulation is kind of like a prediction. He stated that the microorganism must be found in abundance in all organisms suffering from disease, but should not be found in healthy organisms. Essentially what he was saying here is, if a microorganism is causing a disease, then you should find that microorganism in diseased individuals, and you should not find that organism in healthy individuals. Now that makes sense, but that's only because we know now, after a few centuries now, or 1860s as we say. So after lots of modern science, we know that, that makes sense. But this was the first real exploration into this area. So that makes sense. If an animal has a disease, we should find a microorganism that causes that disease in the diseased animal, and we shouldn't find it in the healthy animal. And that's what he found in his experiments. His second statement was the microorganism must be isolated from the diseased organism and grow in a pure culture. So you can see we can now extract that microorganism, and we should be able to grow that microorganism in an agar jelly plate, for example. And we shouldn't be able to do that with a healthy individual because the microorganisms don't exist. And he proved that as well. The cultured microorganism should cause disease when introduced to a healthy organism. So this is really important. So say we've isolated the microorganism that we believe is causing the disease. If we grow that up in a culture, on an agar jelly plate, for example, then I take that culture and infect a healthy individual, then that healthy individual should contract the disease because the microorganism is causing the disease. And number 4, the microorganism must be re-isolated from inoculated diseased experimental host and identified as being identical to the original specific causative agent. So that's quite wordy. So essentially what he's saying here is, now that you've made a healthy animal diseased, when you look at the microorganisms, it must be the same microorganisms that you infected it with that are causing the disease, not something different. So you need to take the microorganisms back out, culture them, and they must be the same ones as you infected the individual with. And that's to make sure that the individual didn't die of something else. You're being absolutely clear. To an extent, you're controlling the variables that could cause death, okay? Because animals can die of a lot of different things. What he was trying to prove here was, yes, in fact, the disease was definitely caused by this microorganism, not something else. Okay, so these were tremendously important revelations and experiments for the development of germ theory. So why is that important today? Well, that is still important today, well, particularly in this time where we're teaching online because a disease is spreading across the world. COVID-19 is spreading, and we think it's mainly through contact with other individuals who are infected, and it's highly contagious. So we know through germ theory that diseases are caused by these microorganisms. And we know that the virus that is spreading across the world just now is causing COVID-19. And COVID-19 is a respiratory disease. And the virus itself is not alive. That's something that, is really interesting. The virus isn't a living organism, but how it interacts with our body can cause untold damage to our respiratory system just by being the design it is. So germ theory is important because it established the cause of many diseases, which led to the prevention and treatment. It is one of the fundamental concepts of modern medicine, and it's saved countless millions of lives. So imagine this hadn't been discovered, and in this day and age we still believed miasma theory was how disease and infection spread. We would think right now that COVID-19 was only being spread through the air because the air was bad. It was like, disease-ridden air, instead of the fact that we wouldn't be washing our hands really. We wouldn't be looking for vaccines because we just believe, it's just in the air. What can we do about that? So yes, it might seem obvious now that we've had modern medicine in our lives and for our entire lives as a society, but societies that came before us didn't have that luxury. So it's not very easy to sort of overstate how important this was. Okay, then I have my A4 fact sheet, the last part of my assessment, and you'll have this in your workbook. You'll have a blank version of this, and you can see on the left-hand side, I know the writing isn't fantastic, I had to try and squeeze it all in here, but the fact sheet has questions that I have answered on the right-hand side of the column. So I've listed my resources, okay, so you can see the web addresses here for where I've got my information, so that's important. In a report, we'd call that our bibliography. You might have done this already in English. Outline the scientific law or theory being investigated. That is, if you were to explain the law or theory to other students, you must be able to logically and concisely outline what the law or theory is about. So this is my really short definition. What is it about? I've got my explanation here. The next question is, what is the experimental evidence or observations? And I've explained my PowerPoint presentation in more detail here. Okay, so I showed you the diagram of the experiment where the top of the flask was kept on or broken off. So I've explained that in more detail here. Assess the evidence that supports it. So how many experiments? So essentially, this A4 fact sheet is working as a sort of a template. So I'd advise you to work on this as you're doing your PowerPoint presentation. And this basically just gives me, say you have a diagram in there and you've just put a diagram in. That's all well and good, but I need you to be able to explain to me what that diagram really means, okay, and why it's important. Okay, so this is the fact sheet that you'll hand in with your PowerPoint presentation. Okay, so that's your assessment task. As I said, you'll have plenty of time to do it in class time. If you have any questions, please let me know. Hamish has another fact. If you open your eyes in a pitch black room, the colour you'll see is called Grau. I think that's German. Sounds German. You see that kind of weird black. I see a kind of black grey colour. It actually has a name. Grau. There you go. Thank you guys.